Okay. Okay. I think I should be good. So whenever you're ready, you can start, I guess.
I will share screen then.
Sounds good.
Okay. So, hi everyone. I'm Mark. So I work at the University of Sydney, and about a couple of years ago now, we wound up accidentally interesting sort of project. We kind of nearly violated a patent, and so wound up on an odd little like path of trying to get around this patent. And in the end, it ended up being quite cool, and things worked much better than we ever expected them to. So I'm going to give you a brief rundown of how it came to be, what we ended up doing, and the sort of various stages that it went through. And hopefully there's some lessons, or maybe you just find it interesting. And if not, hopefully the pictures at least are nice and pretty. So the title is Marketing 101, use the word sex to get people's attention. I will explain what exactly that means if it doesn't make sense later on. So let's get into it. So this all started with a project from an industry partner. So they came to us because they heard that we might be able to take these food poisoning strains, you can see in the bottom right, and make them fluorescent green. So the question that I always get as a follow-up whenever anyone asks at a party, what do I do? Why would you want to make fluorescent green pathogens? The company that we were contacted by, BTF, they're local here in Sydney, but they make these things called bioballs for bioMérieux, global diagnostic, has their fingers in all sorts of different pies about everything to do with life sciences. And the idea is that little pellet in that vial, you can resuspend it and know that there are exactly like 50 plus or minus 1 salmonellas or whatever, and they make them in different sizes. So if you're running, say, a chicken factory and need to be able to show that if there was salmonella getting through, we'd be able to detect it, or that your disinfection process is working well, it's no good to just be like, hey, look, we've got a plate with nothing on it because anything could cause that. You need to be able to say, we put in this many, and even at this many, we can detect them if they're there, or we can get rid of them. And the reason to make them green is that you know that if they're green, they're the ones that you put there, they're part of your test, they're not just something else that happened to get through. So the idea was to make something stable, integrated into the chromosome, and nice and bright. So we had a lot of experience in our lab just playing around with superfolder GFP. It's a pretty classic one, nice and stable, nice and bright. And we'd had a lot of experience with it through the iGEM team that we've been running in our lab for a few years. We're not doing one this year, but we've had an iGEM team for some years now. And the whole iGEM ethos is open source, everything's, available for whoever needs it or wants it. So we thought, great, we can use this one, it'll be fine. Then I think the lawyers on the company end realized that everything was not fine. So this is the patent from a few years ago now, I forget the exact year, but it's still valid is the main thing. And this is everything that they claim. And you can see that it's pretty extensive, but the important one is the first claim. So we kind of need it to be fluorescent. Can't really get around that, that's the whole point. And B is these are 5 particular mutations that they found that take it from the original protein that was isolated from jellyfish back in the early 90s, I think. That's frankly not very good or very useful. And it makes it nice and stable, makes it able to survive at 37 degrees, whereas the old one would turn into, an omelet at that temperature. Makes it nice and bright, makes it nice and stable, means that the cell can make heaps of it and it won't do anything weird. So we kind of wanted to keep all of those mutations as well. But we figured that, okay, 80% identity, maybe we can play around with that. So that's 80% identity at the amino acid level. And that's really broad. Like, I've looked at a lot of patents since we first started this project, and this is about the broadest claim I've ever seen. maybe they got in before people realized just how big a deal this would be, or maybe they just had really good lawyers. But trying to change 21% of a protein at the amino acid level, like you can't even just fiddle around at the DNA level and not change anything in the protein. You have to actually go in and change one residue to another. Any one change could theoretically tank the protein, make it completely non-functional. And I had to find somewhere to put in, I think it turned out to be 52 changes. So that was fun. So this all took place over about a week where I nearly went crazy. But step one was I found every protein sequence that I could that was in any way related to GFP. So this is the original GFP, various variants that people have made over the years, things that are from other species and kind of related, and all sorts of different things. The idea being, if something is the same across every single one that's been isolated, it's probably a good bet that that's important. Don't want to mess with it. But if something is different from variant to variant, that probably means that it's not super essential. And you can probably get away with changing it, or at least that was the hope. So the way I actually went around about finding like 50-something mutations to make. So here's a little example. So I don't know the expertise level of the audience, but hopefully you can see that N and Q are pretty similar. They're both polar but not charged, reasonably medium-sized side chains. And so if something has an N, maybe I can get away with putting it to a Q. And likewise, changing a lysine to an arginine, they're both positively charged. So whatever that positive charge has in terms of the structure and function of the protein, it will hopefully be relatively insignificant if I change that. That is definitely not true 100% of the time, and possibly can screw things up, but I figured, okay, if it's not always this particular amino acid in different proteins, maybe this is a place I can sneak in a change. So the really not at all conserved doesn't look like it matters what's there ones. Those got me the first, I don't know, 20. Ones like this, where it's like, well, we can probably switch this to a that. Doesn't look like it's super conserved. Those got me about another 20. To get the final like 10 or 15 to get over the line, at that point, it was just sort of a crapshoot, like, oh, screw it, put a tyrosine there, see what happens. And so built this horrendous looking spreadsheet, which I'm actually very proud of. So it takes these sequences at the top, just puts each residue in a different box going along, color codes them, because with this many letters, it's just impossible to actually read all of them. I needed to just be able to think in terms of purples and blues. And then over on the right, it actually scored up the differences from the pattern sequence and gave me a percentage, so I would know when I'd gotten far enough that I could stop. So yeah, this was a kind of crazy week. Went a bit off the deep end. It all starts to blur together, and you start to see through the sequences and start reading patterns into them, and it all gets a bit unsettling. But eventually, I wound up with 4 sequences. So thought, okay, I should probably check that I haven't accidentally wandered into a different pattern, because there are a few different ones that claim different things, and you would think, oh, I'm in the clear, and oops, don't want to do that. So turns out, BLAST, there is actually a database in there that is just for sequences that appear in patterns. Annoyingly, it isn't specific to sequences that are claimed in patterns. So it turns up everything that happens to be in a patent. And there are a lot of patented inventions that use GFPs and YFPs and things like that. So I had to go through and check a whole bunch of them just to make sure that I hadn't accidentally wandered into anything. So I don't have any legal training. I did mock trial in high school for a couple of years. So I was a little worried that like, I'm pretty sure I know what all these patents mean, and you start to get a sense of the terms of the trade, but it was pretty confident that by the time I'd put in, 52, 51 mutations, like, okay, I'm pretty sure that by the letter of the patent, I'm pretty sure I'm in the clear. this is all, none of this is to say, by the way, that I don't believe in patents or I don't respect the intellectual property of the people who originally made this thing. They did some very neat experiments, very great science to develop this thing. At this point, it was partly an exercise in, can we even do this? We had a couple of backup options, which were taking unpatented other GFPs and fluorescent proteins from other species and trying to make them better. That was going on in parallel. So my supervisor, Nicholas, Associate Professor Coleman, was taking care of that side of things where I was given this fun task. So you can see on this, these are the 4 sequences that I designed. So this is all done pretty crudely, like by hand in a lot of cases, well, not literally on pen and a paper, but reverse translating the sequences that I designed back into DNA so that we could order them up as G-blocks. And you can see on the little heat map thing at the bottom, there's a few places where I was really certain, like, okay, this looks solid. This is a change that I can get away with. But that doesn't account for most of them. There's a few other positions where 3 out of 4 of them, I'm like, oh, I'm pretty sure I can change this one. And then there's a few where, like, if you look halfway through SP2, there's just a big chunk where I decided to just take a whole bunch of things and say, like, to hell with it. I need to put them somewhere, so I may as well put them here. So at this point, I wasn't really expecting any of this to work. It just seemed absurd on the face of it that you could mutate something 50, like 1 in every 5 amino acids and not have it go terribly. But in any case, forged ahead. This is the setup I was using to actually screen for whether things were fluorescent. We've got an old UV lamp from somewhere, some filters made out of various dyes around the lab and petri dishes, or some cellophane, and an old pair of 3D glasses from a movie, and some LEDs. And this is my dark room, such that it was. That's a towel left behind by someone, a former PhD student in the lab, and using that as a shade curtain. But when it got to the cloning, things, as kind of expected, didn't go so well. You can see SP3 just barely fluorescent. The other 3 completely non-fluorescent. This wasn't the cloning failed and I didn't get it. This was, I got it and it just doesn't work. So it was looking pretty grim, and we were going to just have to go with a couple of those unpatented and spontaneous mutant ones that are there, which were our next best option. But around this time, I think I was just sort of reading old papers about GFP and happened across this concept of DNA shuffling, or sexual PCR. The sexual here comes from the idea of in sexual reproduction, how you have crossing over in the gametes in meiosis, high school biology. And the reason for sexual reproduction is by mixing and matching genes from 2 different parents, you get a lot more variation in your population than one clonal line, and that makes you more resilient to environmental changes. It's, pretty tried and true in the kingdom of biology, but this idea can be extended to molecular biology. So this is, I think, quite a nice, elegant figure from this particular paper, which was my go-to reference for this whole process. So if you think about the sequence space as just like, the entire universe of possibilities, if you're starting at the black circle and you want to get to the star, if you make random changes, or one change at a time that you think might work, you're just going to be going in a little random walk around where you started, and the odds of you actually stumbling across the function you want, or whatever it is that you're aiming to get, not particularly good, depending on how lofty your goal is. The idea on the right with DNA shuffling is that if you take a bunch of different starting options and then take basically mixes of all of them, like if you're crossing them, your possible set of outcomes is so much broader that while 99 point however many 9% of them are going to be complete garbage, the odds are sooner or later one of them is actually much more likely to happen to be the thing you're looking for. So if you've got a good way of screening through thousands and thousands of outcomes, and in our case, it was really easy because all I had to do was look for the green ones. Other papers, like if you're looking, they've made really effective antibiotic resistance proteins for reasons, kind of to prove that it works. And you can, in a couple of generations, make something 10,000 times more resistant to penicillin through this method. So this is not a new process, but it's based on an idea that I have come to really value in biology, is that none of us know what the hell we're doing. Biology it's a black box in a lot of ways. Like, we know a lot of amazing science has been done, but it's ultimately still a bit of a mystery. Like, you never know what's really going to happen. Even one little change can have completely unintended effects. It might do something you weren't expecting. It might destroy something. So embracing that sort of randomness, being like, throwing up your hands and being like, I don't know, let's just make a bunch of things and let the bacteria sort it out. Sort of an approach that I quite like and have come to really value. But yes, as I say, this isn't a new idea by any chance. In fact, I'm pretty sure I got the original idea by reading about some of the very first mutants of GFP. So this is a paper from 1996, and they shuffled GFP back when it wasn't even standardized to call it GFP. You can see there in the abstract, it's called QFP. But by shuffling just the same thing against itself, kind of with just random mutations from error-prone PCR, I think, and doing a screen on it with a visual screen, kind of like I ended up doing, they wound up making something quite a lot better. So this wasn't a completely out of left field idea to apply in our case. The big difference between now and 1996 is synthetic DNA is just orders of magnitude more affordable. Even when I started in biology, like probably 5 or 6 years ago now, a G block would cost, you know, I think it was like a couple $1,000 for like a 1 or 2 KB fragment, or at least several $100s. Like nowadays, that price is down to under $100, I think. So it becomes quite reasonable for even a small scale project to actually order up synthetic DNA. And that's no longer really a limit. And by ordering up synthetic DNA, you can put in changes that you think may work, may not. And the great thing about the shuffling is if you make 52 changes in a protein and one of those nukes it, makes it completely non-functional. But if you can shuffle that against a bunch of other variants that don't necessarily have all of the same changes, sooner or later, one of the variants that you get will shuffle out the specific ones that are bad and leave in plenty of the good ones, and maybe shuffle in some other good ones. And you have so many variants that it's a good chance that one of them will work. So for context, this is what I was like in 1996. So this is not a new process. And I'm not even the first one to do this with fluoroproteins in the modern age. So this is actually done at Stanford by, who I understand you guys, one of your supervisors, Drew Endy, and a group at Atom Biosciences, where they did basically what I should have done if I'd known this was the way things were going to go, and ordered up a bunch of synthetic DNAs of various different fluoroproteins they found in the literature, shuffled them against each other, and then by screening for various colors with facts, I believe it was, you can get all these different colors, and they have fun Christmas or chili pepper themed names. And it's like, I really like that. But I had my box. So this is how it turned out for me. So the process in a bit more detail, you chop it up with DNAs that just randomly chops at various fragments, various sides. So you can see on the gel on the left, starting with quite a lot of plasma DNA going in, after 3 minutes with a tiny amount of DNAs before you stop the reaction, already everything's chopped down to in the realm of 100 base pairs or so. So you chop it up, but all of these fragments are different sizes, different places. And then you heat it up, and in basically a PCR without any primers, where two fragments happen to be similar over a short region, they will stick together, and then your little PCR enzymes will come along and extend out from the end of one fragment, copying based on the other fragment. And you repeat that process overnight. It's a very long PCR, but at the end, you wind up with all these chimeric Frankenstein variants. The ends get a bit messy, but you can go and do a second PCR just on the inner part that you really care about, which I'm lane 4. And then clone that in the same as you would for any normal restricted digest. And so I decided to try this on a whim. Read the paper and I'm like, oh, it actually sounds pretty doable. We have all of the reagents for that in the lab already. They're just PCR enzymes, a bit of some DNAs, a bit of calcium, like basically nothing. If you've got a thermocycler and normal lab supplies, you can do this. And so I did it in just a weekend. I asked Nick, like, oh, do you think I should try this? And he's like, oh, it sounds pretty risky. Probably won't do anything. Don't waste your time. I'm going to do it anyway. And good thing I did, because God damn, it actually worked. So over on the right, you can see this is what the results came out looking like. The things going in were barely fluorescent at all. But when I cloned them into the cells, it actually gave me a decent number of bright green colonies. So I don't know the exact number. I never actually counted them, but looking at across 10 plates like this, that's thousands and thousands and thousands of colonies. Each one of those plates would have 1 to 200 colonies on it. But I can look at all of them quite easily, because all I'm doing is just sitting there with the UV lamp next to my eye, which was not a good idea in retrospect. And I picked the brightest one from each plate, did a little squiggle to try and isolate the colonies, because a lot of them are quite close together, and then measured the fluorescence. And some of them actually worked out pretty well. So the superfold GFP is in there for comparison, but some of them were as bright or even appeared brighter. This number 12, that's a bit of a false positive. 12 is on the bottom row, it's the yellowish one, third from the left. And so because it's more yellowish, it's just an artifact of the way the thing was measuring the fluorescence. The region it looks at in the output from the GFP is actually a bit more yellowish. And so something that is more yellowish sort of cheats its way into looking really good. So there's a lesson there in being aware of the limitations of your methods and sort of questioning things. But a lot of the others looked pretty good, so I sent them off for sequencing. And by looking at the sequencing, I could actually go in and trace the footprint of like where each part came from. And the crossing over really was pretty extensive. You can see like in number 15 here, there's what, 10 or 11 different like identifiable fragments that were pieced together from all these GFPs. However, when I actually counted up the differences, when I plugged them back into my spreadsheet, remember 80% is the line that I need to get to. So this was a painful day to get so close and fall just short. It was a tough afternoon, but realized that a restriction site, the enzyme we happen to already have just in the freezer, a couple of them had more differences on the front half of the protein, and one had more differences on the back half of the protein. And, you know, both were nice and functional. So I thought, well, if I just stick the front half of this one with the back half of this one, maybe it will actually work. And it actually did. It didn't screw it up. GFP is a really nice protein for doing this because it's quite simple structurally. It's just a little barrel with the fluorescent core in the middle, and the barrel is mostly there to protect the core from water outside that would quench the fluorescence. So it's a bit more resilient to doing this than a lot of proteins, but still, it's still kind of amazing to me that with 52 individual mutations, I was still able to get something that wasn't non-functional. Maybe we get something as bright, or even under some circumstances brighter than the one we started with. So as a random mutation that just sort of popped up in the middle of all of this, I didn't change this, but this little colony appeared while I was making those last ones. And you can see it's a lot brighter. And I'm like, wow, this is amazing. I'm going to take advantage of this. And when I went and sequenced it, turns out I'd accidentally recreated something that nature had done, you know, millions and millions of years ago. So this little triad of 65, 66, and 67, that's the actual fluorescent core of the protein. That's the bit that when the protein folds up, it actually reacts with itself and makes kind of like a little fluorescent dye looking bit in the middle of the protein. And so the original one from the jellyfish has a serine there, but way back, some of the first variants, partly because they were looking to do this, partly because it's the way that they were measuring it. But similarly to how my slightly yellowish one looked really good, if you're measuring with blue light, the original GFP does absorb blue light, but it actually absorbs long wave UV, so like just the other side of violet, much more efficiently, and so appears much brighter. That's not particularly useful if you're doing, say, fluorescence microscopy, because those wavelengths give you a lot more background fluorescence in cell culture media. But for my purposes, where the whole idea was to get something that would be nice and bright under just a little simple handheld UV torch, this actually turned out to be pretty fortuitous. But the other thing is, in the intervening years between people switching from the serine to the threonine, that makes it more absorbing under blue light, people have found out a lot of other mutations that make it much better. So that position 64, F, phenylalanine is the original one. So people realized relatively early on that if you change that to a leucine, it actually, that's one of the key ones for making it stable at 37 degrees. So there aren't many variants. In fact, I'm not sure if I found one where people have that serine from the original GFP with those modern stabilizing ones. So this was fortuitous. Like, this was not one of the changes that I tried to put in, but turned out to be all the things that I needed it for, really quite good. So after all of that, it did actually seem to have some things that worked quite well. So this is under blue light, and you can see that a few of them are just as good. One or two of them even seem to be slightly better. But the 315 UV, it still works under blue light, but under UV light, it actually was far better. And that's because its little fluorophore, the core of the protein, is in a state that absorbs UV much better than a normal blue light receptor GFP would. So that was amazing. And yeah, still can't believe it worked. But, you know, it was just over the line, and like had these sort of, you know, recurrent anxiety moments of, what if like lawyers come to me, like the people who hold the original patent are Los Alamos Laboratories, you know, this is the US military we're talking about here. I don't want to be on the wrong side of them. So I wanted to be confident that it was, okay, not just technically over the line, but pretty solidly over the line. This is all still very much like a bit of hanging a lot on technicalities here, but I'm pretty confident that, okay, the patent says this, I do not violate this patent. Claim 1 does not apply to me. So the GFP that I had made was 80%, less than 80% identical to the sequence nominated in the patent. But the sequence nominated in the patent is not actually the superfolder GFP. That one has, you know, those 5 extra mutations. So I wanted to see if I could get it to be less than 80% identical to that one as well, just to make it that much more certain that if I had like one random mutation, it wouldn't accidentally go back into violating the patent. So a nice way to do this was to look at those variants that I sequenced. So FP1 to 4, those are the ones that I actually designed and put into the shuffling mix. So there's 2 little bits I'm using as examples here. So over on the left, 110 to 112, you can see that 3 out of the 4 of them, I was like, glycine, what's E? Glutamic acid, isoleucine, that's pretty safe. I can probably get away with that. That seems nice and safe. But only one of them had the original AEV. But if you look at the actual variants that I pulled out, none of them have that GEI. All of them have the AEV that was only in one of them. So pretty solid bet that G and I, one of them is not a good thing to have here. Whereas over on the right, 142, you can see that only half of the variants that went in had a D. The other half had the original E that's in the patent sequence at the top there. But of the 5 that I sequenced, 4 of them had the D. So I'm like, well, if, you know, less than half going in have it, but, you know, 80% coming out do, then that probably suggests that it's good, if anything. So this is not the most robust analysis. Drawing conclusions off just 5 variants, like I really should have sequenced more, but those are the ones that I sent off because they were nice and bright. But it says that, okay, there are some changes that you can see that D isn't actually in the 315 UV that I was going with at this point. So I was like, okay, maybe I can change that. and get a few extra changes compared to the patent. And who knows, it might even make it brighter. Or at the very least, it seems unlikely that it will screw it up. So I identified, I think, 6 different regions, some with a couple mutations that might have this effect of like being able to just squeeze in a few more and get it over the line. So did that, and you can see a few different combinations there. Some of them turned out to not be very good. So you can see C there is a marked step down from the original. And some of the ones that are on the right where I've combined several different changes, they're still fluorescent, but they're not very good. Whereas the E and F versions actually made it a lot better. Partly, I think there was some issue with different strengths of promoters in the test that I was using. But in any case, E turned out to be really bright. And so that's the one that we finally named free use GFP or FUGFP. And that's the one where if you go and get the sequence online or from AdGene or from Nick's Twitter, that's the one that it will be. And yeah, so it has 54 differences from the patents, 48 from Somerville GFP itself, and it's nice and bright. Seems pretty stable from all the tests that I've done. Still haven't done the final few experiments to get it published because the priority is still the actual original project with those industry partners from way back in slide 3. Those are the people who pay my bills, so I have to get that done first. And it worked really well for the first 3 or 4. The salmonellas, the food poisoning E. coli, those are nice and green. The 2 listerias, those are a pain in the neck. I'm still working on them. I've been through a bunch of false starts and resets, but it's nice and green. So we will get that paper out sooner or later. But still, you know, at this point, I'd read a lot of papers about GFP and figured I knew a thing or two, and I couldn't resist the chance to, well, let's see if I can make the different colors. So from the original green fluorescent protein, by various random mutations people had found over the years, you can push that one way or the other. So green becomes yellow. You can make it blue. You can change the absorption properties. And these all come down to pretty specific changes. So this is all in 1 or 2 or 3 spots, changing this to that, like either right in the core of the fluorophore or a couple of sites that are elsewhere in the sequence, but when the thing folds up are all in the same place, right in the center, and that's pretty profound effects. So realized that if I had degenerate primers where the actual primer that arrives via PCR has a mixture of different options at some of the bases, you can get all of these different amino acids to come out in a single experiment. So I really like this plate because 1, it behaved exactly as I'd hoped it would in that 1 cloning experiment, I get all these different colored variants, and 2, I just think it looks pretty. So this is that serine that I mentioned earlier, is the one that's in the original one that makes it more UV sensitive. So if you change it back into a threonine, it behaves much better under blue light. So over on the left, you can see those ones that are quite fluorescent under the blue light, even without filtering. Even with, this is another very low-tech piece of lab equipment here. Our blue LED light source had broken at some point. So you can actually see it reflected in the surface of the plate. But that is my phone with a full screen image of Google image search for the word blue. Didn't have a blue actual blue light source, so that's just my phone with blue on max brightness as my light source, which wasn't great, but it worked. Whereas if you change the one next to that serine to a histidine, you wind up with a blue fluorescent protein. It changes that core fluorescent region just enough that it absorbs UV still, but instead of putting out green, it puts out blue. And it looks really pretty. Elsewhere in the protein, so there's a threonine in position 203 that folds back to be just next to that fluorophore and interacts with it. If you change that to an isoleucine, it actually completely abolishes its ability to absorb blue light, and it only absorbs the UV, but still gives out green and gives out really brightly. Whereas if you change it to a tyrosine instead, it actually shifts the absorption and the emission wavelength up towards the red end, and you wind up with a yellow fluorescent protein. So you can see under the blue light, it's a much better blue light source by this stage. Some of them turn out really fluorescent and much more yellowish looking. And just under white light, you can almost see that dark yellow color, which if you've got any experience with E. coli, you know, it's not normally that. It's much more of a creamy beige color. But I wound up with a couple of different yellowish looking things that kind of looks like a 70s wallpaper print to my mind, those colors. And then, yeah, at the top right, you can see this is work that I've done purifying these proteins for experiments. And so now I have a little box of incredibly unnatural looking bacteria juice sitting in the freezer. And you can see a lot of them have slightly different colors. Some of them are very different colors, obviously, with the blue one. And over on the right, that's the original super folder. So this FUGFBB is spectrally, in the way that it responds to light, pretty much identical to the super folder GFP and eGFP and most of the ones people use these days. But this way we've got sort of a different set of options for if people want a free use intellectual property free GFP, they can, but if they also want one that's blue or yellow, we can give that to them as well. And the advantage with doing it this way is that it still behaves much like a traditional GFP. Like in every respect that I've seen, like I haven't found anything yet where it's like, oh, it's not like the original GFP when you do this to it. The only thing where I would expect that it wouldn't be the same is if you were doing something with anti-GFP antibodies. I haven't tried it, but I wouldn't be surprised if your anti-GFP antibody that you buy doesn't work for this one, because antibodies are pretty specific, and I've changed 20% of the protein, and the majority of those changes happen on the outside where it's not affecting the fluorescent core. But in every other respect, it would seem to be pretty similar to the original one. So you could sort of plot it out if you had been working with a patent protected GFP, which was fine for teaching and research. But if you wanted to spin that out into a commercial thing and suddenly realize, oh no, we don't want to pay royalties, this is something you could slot in much more easily. So the takeaways from this, the things I've learned, and hopefully you may have learned, it's not a synthetic biology presentation until you've included the design, build, test, learn cycle. And that sort of didn't actually set out to apply this sort of framework to it, but it's really what we did. Designed something, built it, tested it, didn't work, learned from my mistakes, like, okay, maybe this wasn't such a good idea. So designed in a few more changes, built that, okay, it's working better, but we can probably learn a few more things, designed those, built them again, tested them again, and gotten a few times around the circle at this point. But overall, like the lesson I think here is to be flexible. None of this turned out how we originally planned. There's so many different levels of, oh no, we're gonna have to change things. And it actually, as long as you're able to roll with the punches and be like, okay, if we can't do it this way, maybe we can do it this way. If this doesn't work, thinking about like, okay, how can we maybe make lemons out of lemonade? So those original variants that I made were all garbage. None of them were any good. But by shuffling them, I was able to, filter out the parts that weren't very good, and wind up with something that's actually, pretty great. Embracing the uncertainty, like I was saying earlier, it's like no one knows what the hell they're doing really. Deep down, like it's all a bit of a mystery. We know a lot. I certainly know a lot more now than I did at the start of this, but you can't be certain of these changes. So making individual changes on the hope that it will work as you expect is just sooner or later you're going to run aground. So blowing up your sample space and like the law of very large numbers, even if something's 1 in a million, if you screen a million variants, then it's a good chance things will actually work sooner or later. So as long as you have the ability to screen lots of variants simultaneously, and not have to pick each one and actually do a whole test on it, like if you can just screen thousands at once, whether that's with a flow cytometer or just on a plate with a UV lamp with a towel over your head, that's a great way to wind up with things that you wouldn't have gotten by using just the traditional linear process. Old doesn't necessarily mean outdated. These processes, like nothing I did here was super cutting edge. Like I didn't CRISPR in these changes. The synthetic DNA is about the only novel thing about it. Old here is still, we're talking about the 90s, but there are processes from the, I find myself reading papers from the 70s and 80s surprisingly often, and sometimes those old methods, like, they worked, and you don't need the latest high-tech piece of kit or expensive reagents. Like, they're old school methods, but they work for a lot of things. So don't be distracted by the flashy and the new. Sometimes the old way does actually work really well. Sometimes it's terrible and you should use the new method, but being aware that there are lots of different ways to get to the same end, and sometimes other things work well. It's good to have a broad toolkit for, hey, you never know when something will break and you'll need to use something old, or there'll be a global shortage of your reagent and something else that's been sitting in the fridge for 10 years will actually be just as good. Yeah, learn from your mistakes, have humility. If you take every failure as like, oh no, I'm terrible, I'm bad at science, maybe this just isn't for me. Like, no one survives in science by just having win after win after win. Everything is going to fail eventually. And the only thing you can do is learn from those failures, take them as like, okay, things didn't work, so they just haven't worked yet, or what can I learn from what went wrong here? There are changes where if I look back at those 4 original variants that I designed, like, what was I thinking? Why didn't I read this paper and realize that that was a terrible idea, or put in this change, which would have been, you know, a great idea from the start? I'm pretty sure I know why 1, 2, and 4 all failed, and it's because I based them on a particular variant that was just not a good idea in the first place. So now I know a lot more, and if I were to do this again, I could do it a lot better. And I talked to the iGen team or Biomod a couple of weeks ago, and I sort of, there are a lot of things that if anyone wants to know, I'm more than happy to share my ideas to how to do a project like this much better, and the things I would do differently if I did it again. But you get that way by making those mistakes, just trying things, having them not go well, and learning for the next time. And then sometimes the next time it actually turns out to be pretty great. So thank you for listening. Please no one sue me for this. This was something where it was like, oh no, we don't have much money. Our industry partners were like, hey, you promised to do this for us, and the thing you promised to do is going to be a bit of an issue with royalties, and like, it was all a bit stressful, but it all turned out pretty great in the end. So I just want to thank Nick, my supervisor, that's him in the middle there. You can follow him on Twitter, Colmenomunis. All his top tweets are my stuff. So I don't have a professional Twitter to share here, but if you want to see what we're up to and what I'm up to, that's probably the best social media thing to do. But the whole lab, because even if people aren't directly giving you advice on your project, there's so much to be said for just having people who know what you're going through of like, oh, my experiments have failed again for the 6th week running. And I don't think you can survive in this kind of science without people to just vent with or have a consoling cup of coffee with. So major thanks to all of them. And big thanks to our industry partners. They've been very patient with the whole, like, just give us a couple months to get like make a whole new GFP for you. And yeah, they've been great with supporting this, and they're quite happy with us talking about this work that we've done. It's not all like, oh no, you have to keep that secret. And they've been really great to work with as well. So thanks for listening. At this point, I think Chris, are you in charge of like questions if we've got time for it?
Yeah, that works. Yeah, I think yeah, if anyone has questions, they can just kind of drop them in the chat, or even like unmute, I guess, if you're feeling comfortable, and just ask them.
Yeah,
Yeah.
it looks like we have one question in the chat already, which is it's basically asking about like countries that patents are in, and kind of the ones that you had to deal with.
Sorry, one of my earphones is going flat, so I'm just trying to charge it. So hopefully, can everyone still hear me fine?
Yep.
Okay, great. I wonder if I can see it if I go to the original patent. Yeah, so the one that I took a screenshot of, it says United States patent. But when you go and look at the patents, and you can see these, Google's got quite a nice patent viewer. They come in families. So this one ties to a bunch of worldwide ones. And in any case, like the products that bioMérieux is going to be selling, these bioballs, it will be sold in the US. Like that's a major market for these things. Like in fact, I think it's the primary market, because the US has much looser regulations on genetically modified pathogens being handed out to people. I have a feeling, I think one of the industry people said to us once, and no one quote me on this, but I have a feeling in the EU, I'm not even sure this thing would fly. Like the restrictions there are much stricter. So the US has got obviously the biggest market for these things. And so the United States patent is the one that really mattered to us anyway. So even if it was only valid in the US, it would still be an issue. But usually when people apply for a patent like this, they also wind up with patents that have much broader jurisdiction. Like it would be very unlikely to find something that's only patented in one country and otherwise completely up for grabs. And in any case, that wasn't really a line we wanted to try and tiptoe around. We wanted it to be pretty unambiguously. It's like, okay, even under the broadest possible claim, our thing still comes out on its own and not under anything else. So yeah, so there are so many patents out there, but while the United States one is the one we were looking at here, yeah, you can't really just say, oh, it only matters for this country and we can ignore it. You kind of have to treat them as pretty universal in that sense.
Yeah, another question we have in the chat asks, what does one use the GFP antibody for?
It's not something that I do much of, and like the kind of work that I do, but I know that especially people who do cell culture stuff, that they're looking at, you know, putting things into cell culture lines and saying, oh, what does it do? Like often you tag something with GFP to follow your protein of interest around the cell, or, you know, if you're working on a virus and you want to see where that virus is going in the cell, if you stick a GFP on the side of it, you can follow it in real time under a microscope. Like the lab we work in, is shared between a couple of different microbiology groups, and the people 2 benches up work on vaccinia virus, and they've got a whole range of different brightly colored fluorescent tagged viruses, and they can follow it in a very nice fancy $250,000 microscope and see what it does. So if you've already got your GFP tagged onto your virus, or if it's attached to something of interest, if you want to know where that virus is winding up in something, you can do a Western blot and like take the cells, get the protein out, run it out on a gel, transfer it to a membrane, and then you wash over the antibody. And the reason you would use an anti-GFP antibody is you probably can't buy a commercial antibody for your particular favorite virus's random coat protein. But you can buy one for GFP because GFP gets tagged on so many things that there's a market for people to just have an anti-GFP antibody. And so you can use that to pull up any GFP fusion protein that you happen to be making. So I think that's the reason why people would use them. So that's like the biggest drawback I can think with FUGFP is if you wanted to do that, those antibodies wouldn't really be available. But I don't think it's a huge issue because I don't see why, I don't know of any circumstance where you would want an anti-GFP antibody. Or like if you were making a commercial product that needed this patent-free GFP of ours, a situation where you then also wanted the anti-GFP antibody would be less likely. It's something you would do more in a research setting, I think. I'm sure there's exceptions to that, but That's sort of the best guess I have as to where someone would use one of those anti-GFP things. And yeah, I don't think it's a huge issue for us. Who knows, maybe it does work, but it's not something we've tested.
Okay, thank you so much. Another question we had was if you had any insight into how you made that Excel spreadsheet that you kind of showed us on one of the slides.
I mean, I wonder if I can pull it up. It might actually be on the computer. So yeah, like it's not the most elegant thing. It's the here it is actually. So can you see this? Like the screen sharing the Excel,
No,
or is it
it's
just
still
sharing?
sharing on the slides.
Yeah, I think you may have to like
Oh yeah.
unscreen share and then start it again.
Yeah. Give me a sec, everyone. Shift screen to. Yeah. Here we go. Here's my wonderful spreadsheet. Bit of a mess. So this is just copying in the raw amino acid sequence that I got from online. And then there's a lot of hidden rows here. Yeah, so this is, I can't remember what this one was I used. Yeah, so this is the mid function. So take, one of those boxes and go this many cells along it based on the little number up there. And then just pull out that letter. And then, yeah, there's a hell of a lot of conditional formatting going in to color code each of the amino acids. So each one's a different color, obviously, but also things like leucine and isoleucine are different shades of green because they're quite similar amino acids, whereas D and E are both negatively charged. So they're both bluish purples, and likewise for the red positively charged ones. Yeah, so then those changes by down here by saying like, is it the same from this one to the pattern sequence? And then it like counts them up here and then just gives a little percentage. And then down here, these are the ones that I actually made. So here, let's do it at the beginning so it's nice and easy to see. If I change this G to, I don't know, Q, let's see, change the color. And then if you look up here, this one just like stitches them all back together with the concat function. And those were the sequences that I was able to pull out. So it's not the most elegant solution, but it's something that I could do quite easily just in Excel. And it was really, really great for being able to plug in little changes and see like, get a counter going in real time. And the end of that, it's a real solid week of just staring at this wall of letters and colors, being able to just sort of not have to read individual letters, just be like, oh yes, green to yellow to blue, being able to do it visually. I'm a big fan of color coding things for just reading letters. Sooner or later, you're going to screw things up. I think on my bench in the lab, I have somewhere between 40 and 50 different Sharpies in every color I think they've ever made, because I am a big fan of color coding things so that I know that, okay, I'm putting the red thing and the red thing and the blue thing and the blue thing. So yeah, pro tip for not screwing up is give yourself as many different ways of just, even if it's the end of the night, you're really tired, you're like blue to blue, red to red. Yeah, so that's my spreadsheet. And then there's a few more things in this with like, here's the process for adding in those mutations. So here's the little degenerate primers and the different mutations that they put in and the different options. So here's all the possible outcomes of that plate with all the different colors on it. There's actually even more variants than the ones that we've actually pulled out and are working forward with. There's quite a few different things you can get, like different combinations with a few different colors. And then here's some calculations that I did for something else. And this is the actual cloning. But yes, that's my nightmare of a spreadsheet, but I'm quite proud of it. I quite like it.
Yeah, I can only imagine all the work you put in on the spreadsheet. It's definitely, yeah.
Sort of
I
thing where
guess
if I had good coding skills, you could probably make a very elegant thing in Python to do this. But hey, Excel's good. I understand Excel. And yes, I can do some quite fancy things with it, if I may say so myself.
Yeah, it looks good. Another question we had was, what was the best way that you found for finding like relevant patents for kind of for obviously GFP, the one you're working with?
For finding patents.
Yeah, like relevant ones to GFP.
I think in terms of finding patents, the main thing was the blasting the patent database, because I started with these. Well, I started with the original sequences, and then I went and made my own ones, and then blasted them back, and went and read through all the different patents. Like they sort of come back as just a list of US patent, and then like 9-digit code or whatever it is. And I got to the point where I could recognize like, oh yes, this one is this particular protein, because a lot of them, particular patent, a lot of them come up a lot of times. But in terms of actually designing these things, I didn't go by harvesting patents. I didn't say like, aha, whose ideas can we use in our thing? The ideas for where to put the changes, those mostly came from just that alignment and sort of making an educated guess. I'm like, oh, this probably can be changed. A few were finding papers where they're like, oh, this change actually makes things better, and folding those in. But yeah, as far as the patents, you can search patents on Google quite well. And there are a few more specific patents looking sites. It's been a while since I did it, so I'm sort of a bit hazy on the details. I couldn't give you like a URL off the top of my head, for example. But yeah, by blasting them, finding things that were similar and checking that they were not too similar. Sorry, there's a cat yowling at a fancy pigeon outside. That's the same, by the way, a fancy pigeon, a domestic species. But yeah, so in terms of finding the patents, the database was the easiest way to find things that were similar. And then it was sort of just a brute force thing of reading down and clicking on each one and checking that, okay, no, they're not claiming the GFP, it's just the GFP appearing in the context of some other patent, some other thing that was the actual focus of the invention. But yeah, so that's about as much as I can say on the patent. Like, I'm definitely no expert. There are people out there whose whole focus is the intersection of science and law, and are much more qualified to speak on this than I am.
Okay. I guess just a question I had is whether you had any plans to kind of continue to try to make free use versions of proteins, whether it be, I guess, just kind of in the future, whether you'd want to try to tackle this issue, but with a different protein?
I don't have any plans to do that myself on anything in particular. It's something that has sort of become, I wouldn't say a focus, but it's a running theme in our lab. So our iGEM project 2-3 years ago was to make an open source insulin. So it's connected to the, I think they're called open source insulin or something. Can't remember the name off the top of my head, but basically it's an organization to be like, hey, insulin prices are crazy, through the roof. This is a life-saving medicine. There are people who are going without or having to ration their insulin because it is so egregiously expensive, especially in the US. And so that became our iGEM project for that year, trying to make an insulin that could be distributed open source. And I think the people on the call we had a couple of weeks ago with Nick, he spoke a lot more about that. Insulin's a big business. So the university's like legal department kind of got in contact with us and like, you need to shut this part down, like, because a couple of the things that we're working on wound up infringing or looking like they might infringe on some patents. So yeah, that was a stressful week in the lab for Nick. But yeah, so I wouldn't be surprised if, because it's quite a good story. Like, this has resonated with people. Like, this little thing that we were doing is like, oh crap, we need to just quickly fix this on the down low. Like, turned out to be like the most impressive thing I've ever done, certainly. Like, look at me now. I'm talking to a bunch of people on the other side of the ocean at 10 a.m. on a Friday morning. So it's something that I wouldn't be surprised if we wound up going down other avenues. but no, we don't have any specific plans to do anything at this stage.
Okay, thank you. If there aren't any other questions, I'll stop recording and kind of let you go about the rest of your day. But
Rest of your Friday morning.
yeah. Okay, I'll stop recording.
Talking to you from tomorrow.